Hello everyone, my name is Sonu and I am the student of BSc Chemistry Honors 3rd year. Today we will prepare a video for the visually impaired students on the topic of fermentation. Fermentation is the chemical transformation of organic substance into simpler compounds by the action of enzymes, complex organic catalysts which are produced by microorganisms such as molds, yeast or bacteria. Fermentation technologies use of microorganisms to produce food, pharmaceuticals, and alcoholic beverage on a large scale of industrial basis. The science of fermentation is called zymology. Principle The basic principle involved in the industrial fermentation technology is that organisms are grown under suitable conditions by providing raw material meeting all the necessary requirements such as carbon, nitrogen, salt, trace elements, and vitamins. The end products formed as a result of their metabolism during their lifespan are released into the media which are extracted for use by human beings and that have a high commercial value. Type based on respiration. First is aerobic fermentation. It is a metabolic process by which cell metabolize sugar via fermentation in the presence of oxygen and occurs through the respiration of normal respiratory metabolism. Aerobic fermentation means that oxygen is present. Wine, beer, and acetic acid vinegar such as apple cider vinegar need oxygen in the primary or first stage of fermentation. When creating acetic vinegar, for example, exposing the surface of vinegar to as much oxygen as possible creates a healthy, flavorful vinegar with the correct pH. Lactic acid fermentation. Lactic acid is formed from pyruvate produced in glycolysis. NAD plus is generated from NADH. Enzyme lactate dehydrogenase catalyzes this reaction. Lactobacillus bacteria prepare curd from milk by this type of fermentation. During intense exercise, when oxygen supply is inadequate, muscle drive energy by producing lactic acid, which gets accumulated in the cells causing fatigue. Alcohol fermentation. This is used in the industrial production of wine, beer, biofuel. The end product is alcohol and CO2. Pyruvic acid breaks down into acetaldehyde and CO2 is released. In the next step, ethanol is formed from acetaldehyde. NAD plus is also formed from NADH, which is reused in glycolysis. Yeast and some bacteria carry out this type of fermentation. Enzyme pyruvic and decarboxylase and alcohol dehydrogenase catalyze this reaction. Citric acid. Citric acid derived its name from Latin word citrus with a molecular weight of 210.14, which option 3 carboxylic functional group with metabolic product is the most important organic produce large quantity of extensively used in food and pharmaceutical industries. Citric acid production. About 99% of world citric acid production occurs through microbial process which can be carried out using submerged or surface cluster. The 70% of anhydrous or monohydrate form of citric acid is used in food and beverage industry as an acidifier or antioxidant to preserve and enhance the flavor and aromas of fruit juice, ice cream, and the marmalades. 20% used in pharmaceutical industry as antioxidant to preserve vitamin, pH corrector, bulk preservative, or in the form of iron citrate as a source of iron for the body as well as in tablets. ornaments, and cosmetic preparation. In the chemical industry, which use the remaining 10% is employed as a foaming agent for the softening and treatment of textile. Surface fermentation. Using rice bran or other starch or sugar-rich substance have been used for surface fermentation by aspergillus or in the liquid solution in flat aluminum or stainless steel pans. Vinegar can be used to produce citric acid despite the high content of trace metal in rice bran are used. Citric acid is extracted from the bran by leaching and it's then precipitated from the resulting solution as calcium citrate. Submerged fermentation it is the common practice where citric acid is made. The fermenter is made of acid resistant material such as stainless steel. The carbohydrate source are molasses deoxygenized by ion exchange, sucrose or glucose, MgSO4.7H2 and KH2PO4 at about 1% and 0.05 to 2% respectively are added. The pH is never allowed higher than 3.5. Copper is used up to 500 ppm as an antioxidant of the enzyme which require iron 1 to 5% of the methanol, isopropanol or ethanol when added to fermentation containing unpurified material increase the yield. The yield are reduced to media with purified materials.